Hi guys, my name is Jasmine. I'm a PhD candidate at the University of Manitoba in the Department of Medical Microbiology and Infectious Diseases. I'm here today with Soapbox Science to tell you a little bit about my project and what I've been working on for the last couple of years of my degree. So my science career has been really focused on viruses. I think viruses are so interesting. They're so small, but they could have huge outcomes, as we're seeing in today's climate with SARS-CoV-2. The virus I work on is called the Mumps virus. So you may have heard of this virus that often causes swelling in your parotoid glands, so swelling in your neck here. And Mumps is a Paramyxoviridae virus. It is an old virus. It was first described by Hippocrates in the 15th century. And it's kind of an interesting virus. So a lot of viruses will have some sort of reservoir. So an animal reservoir often. So for example, influenza can be found in birds and in pigs. However, Mumps doesn't have this animal reservoir. Humans are its only natural host. So somehow this virus has survived for centuries just by infecting humans. It's also almost like a perfect storm in terms of causing an outbreak. So Mumps has a really high infectious rate. It is just as infectious as the flu. It also has a very long incubation period. So for example, if I were to get mumps, I would be infectious for 1 week before I show symptoms, as well as 1 week after. So that means I would be shedding virus and able to infect other people. So it ends up being around a 16-day incubation period. So here we have a really infectious virus when people who are infected are able to shed or pass the virus along for a long time. This is great for the virus to cause outbreaks. Not so great for people. So let's talk about who mumps infects. So prior to the vaccine, mumps was a disease of children. It was often a childhood disease, most often just causing swelling in the parotoid glands. It's very painful. It can also have some more severe diseases, especially in those who get it as an adult. So you could get things like permanent or temporary deafness, infertility, encephalitis, so swelling in the brain. And actually prior to the vaccine, mumps was one of the leading causes of viral encephalitis. But then we got the vaccine. And the vaccine was great. It reduced cases by 99%. It was really effective. We used to have 12 circulating genotypes of mumps, and to date we have 6. It was a good vaccine. It's a part of often the mumps, measles, rubella vaccine. So that was great. However, in the last couple of years, we have seen a number of outbreaks occurring now, not in children, but in young adults. So you may remember there was some notable outbreaks like the NHL outbreak in 2015 and 2017. I remember I always go back to the picture of poor Sidney Crosby with his unilateral swelling in the parotoid gland. So these outbreaks often are starting in sports teams or universities, places where people are in high contact with one another, because remember, again, it's a very infectious virus, but it's happening often in vaccinated populations. So now we have some new questions. The question is, why is this happening? Is it because the virus is mutated? Interestingly enough, the vaccine most people get in Canada and the US is a genotype A vaccine based on the Jerry Lynn strain. However, the circulating genotype, the one that's causing outbreaks not only in Canada and the US but also in Europe, is actually genotype G. So now there's questions, do we have waning immunity? Or do we have a lack of cross-reactivity between the vaccine and this new circulating strain? Or what's going on? So that's kind of what my project aims to focus. So I have multiple parts of my project, but I'll just talk to you today about one. So one of my projects is looking at the Canadian outbreaks of mumps, and there have been a number of them. Some notable provinces include BC, Alberta, Manitoba, Ontario, Newfoundland, Nova Scotia. And I'm going to look at these outbreaks and try and determine a couple things. First, I want to see, is the virus mutating within an outbreak? So I'm going to use a method called whole genome sequencing, and I will be able to identify the exact genetic material of the viruses within these outbreaks, and I'll be able to see if we look at the beginning and the end of the outbreak, is it the same virus or has it changed? And that will give us an idea if perhaps do we have a vaccine escape so a virus that the vaccine does not have antibodies against that is circulating within Canada. I'm also going to compare these sequences to samples that are archival samples, so samples that are 5-10 years old, and see, is this the same virus or has it changed over time? That again will give us some insight as to whether or not the virus is mutating, and that is what is allowing for these outbreaks to happen, or if it's actually the same virus throughout this whole time and perhaps the vaccine does not have the cross-reactivity needs against this genotype. I'm also going to be looking at whether or not we could identify populations that may be more at risk. So I'm going to be looking at some epidemiological factors, things like gender, geographical location, age, and vaccination status. And the reason this is important is because it will help us identify, let's say there's another outbreak in the future, we will know, okay, let's say people of a certain age in a certain location seem to be more at risk. Public health can then go in and try and isolate those people or ensure that they are not exposed to prevent the outbreak from spreading further. So it'll help inform public health policy surrounding future outbreaks. So my project has a lot of components and I'm still working on it, but I do like it and I find it really interesting, and I hope you have found it interesting too and that this has made sense to you. But basically the end goal of my project, not just this aim but all of my aims, is to help better inform public health policy and what to do with future mumps outbreaks, determine how we could combat future outbreaks. Do we need a third dose of the MMR vaccine? Do we need a new vaccine that is perhaps more cross-reactive against the circulating strain? Right now we don't know, and so it's hard for public health officials to make that call because changing either of those things will take a lot of time and a lot of money. And so we want to be sure before we make that change. So my project focused on mumps, trying to prevent future outbreaks and better inform health policy. Thank you guys for listening. Have a great day.
So, if I tell you that whatever we studied in the high school physics, saying that an atom or electrons, protons, neutrons are the most fundamental particles is not true anymore? What if I tell you that the universe that we see around, including our very own Earth, Sun, all the galaxies, everything that is visible constitutes just 5% of the mass of the universe, and the rest 95% is dark matter or dark energy, which we cannot even see or detect? How would you feel to know that 100 trillion neutrinos, which is also one of the fundamental particles, they pass through our bodies every single second and we can't even feel them or detect them? I'm sure by this time you're feeling a little surprised. So, well, my friend, now is the time when I would say, welcome to the world of particle physics. Hello everyone, my name is Sakshi Kakkar and I am a PhD candidate at the University of Manitoba, Winnipeg. My research is about measurement of lepton-lepton electroweak reactions. Leptons is one of the two basic types of elementary particles which are the building blocks of matter. The other type is called quarks. As mentioned in the Standard Model, all matter around us is made up of these two types of elementary particles. So, Standard Model explains how these basic building blocks of matter interact, governed by four fundamental forces, which are strong force, weak force, electromagnetic force, and the gravitational force. Even though Standard Model is currently the best description there is of the subatomic world, it does not explain the complete picture. The theory incorporates only 3 out of 4 fundamental forces, excluding gravity. There are also some of the important questions that it does not answer, such as what is dark matter? What happened to the antimatter after the Big Bang? My research tries to find answers to some of these questions. Now, to quickly go over what is dark energy and dark matter. The visible universe, including Earth, the Sun, other stars, and galaxies, is made up of atoms, and all this ordinary, or in scientific terms, the baryonic matter, makes up less than 5% of the mass of the universe. The rest of the universe appears to be made of a mysterious, invisible substance called dark matter, which constitutes 25% of the universe, and a force that repels gravity known as the dark energy, and that constitutes 70% of what universe is. Scientists have not yet observed dark matter directly. It does not interact with ordinary matter and is completely invisible to light and other form of electromagnetic radiations, making dark matter impossible to detect with current instruments. But scientists are confident that it exists because of the phenomenon like expansion of our universe, which is accelerating and which is contrary to what one might expect from a universe that began in a Big Bang. So, in my research, we are trying to measure asymmetry of polarized electrons, which is closely related to a fundamental parameter of the Standard Model, which is called weak mixing angle, and this can be tested for new physics related to dark matter, etc. So, how do we measure asymmetry? In the experiment, we study scattering of longitudinally polarized high energy electrons from unpolarized electrons. The polarized electrons have become an essential tool in nuclear and particle physics to understand the fundamental forces of nature. Typically, these studies need longitudinal beam polarization where the electron spin is oriented parallel or anti-parallel to the beam momentum. Electron beam polarization is determined by measuring the difference in the electron scattering rate for the two possible helicity configurations. The ratio of this difference to the sum of scattering rates is called asymmetry and is directly proportional to the beam polarization. The scattered electrons then are focused and guided to a set of detectors that measure the asymmetry in the number of scattered electrons as a function of polarization. So once we know the asymmetry factor, we can find out weak mixing angle, which is a very fundamental parameter in the Standard Model, and that helps to find out new physics beyond Standard Model and dark matter and dark energy. So it gives a good insight into that. So although Standard Model accurately describes its theory, it is still incomplete, or perhaps it is only a part of the bigger picture that includes new physics deep hidden in the subatomic world or in the dark recesses of the universe. So, my friends, this is something to watch out for sure as it unfolds itself. But one thing is for sure, that the future of these particles looks very magnetic. Thank you.
Hello, my name is Silvia and I am a data analyst at the Chronic Disease Innovation Center here in Winnipeg, Manitoba. I am originally from Colombia, where I graduated from medical school and I work as a physician in the emergency department. Medicine is founded on scientific knowledge, and research very often shapes our clinical practice. Although I understood the basics of clinical research, I wanted to go beyond reading about other people's research projects. I wanted to understand about research and become a physician scientist. And that was one of the main professional goals I had for emigrating to Canada. In Canada, my research path started by volunteering in research projects and by doing a graduate diploma in clinical epidemiology. Clinical epidemiology is research oriented to improve clinical and patient-oriented healthcare. Once I finished that program, I was accepted for a master's in community health science. That allowed me to understand the methodology, the study design, and the clinical applicability of the research projects. Fascinated by the kidney because it's an elegant and complex organ, I decided to do my research in kidney disease. And I chose an advisor who is a nephrologist and a researcher and work as a physician scientist at this lab called the Chronic Disease Innovation Center. This lab is led by a group of principal investigators who had a broad array of medical, data analytics, health economics, and research skills that are put together, and we work closely with academia and industry. A nephrologist is a doctor who specializes in treating diseases of the kidney. All living things must remove waste from their bodies. Reptiles, birds, mammals, and other animals remove the waste, some of those waste products through two body organs called the kidneys. In humans, the kidneys are between 10 to 12 centimeters, 5 inches, which is about the size of your closed fist. We have one on each side of the spine. The kidneys are two of the hardest workers and complex organs in the body. The kidneys filter the blood, taking out all the waste products in the blood. In 24 hours, the kidneys filters and clean about 200 liters of blood. Then the kidneys remove that waste and send it to the bladder in the form of urine. They don't only do that. They also regulate the body fluids in the body. They play a role in controlling the blood pressure and the production of red blood cells, and they help you maintain healthy bones. Remarkably, humans don't need both kidneys to survive. You can live a completely healthy life with just one kidney. Kidneys are part or a team of body organs. They work closely with other organs such as the liver and the heart. The kidneys and the liver work together to remove toxic waste from the body. On the other hand, the heart pumps blood that is filled with oxygen through all the parts of the body, including the kidney. And as I mentioned before, the kidneys then clean this blood and remove all the waste products and the extra water. If you are diagnosed with chronic kidney disease, that means that your kidneys are not working correctly to remove the waste and excess fluid from your body. In Canada, 2.6 million Canadians live with chronic kidney disease. That is about 1 out of 10 Canadians. There are two main causes for chronic kidney disease: hypertension and diabetes. To measure if you have kidney disease, we use a blood test. In it, we measure a waste product in the blood that is called creatinine. This blood test, plus your age, your size, and your gender, is used to calculate how much your kidney is filtering per minute. That measurement is called the glomerular filtration rate. With that glomerular filtration rate, we can classify your kidney disease in 5 stages. That goes from 1, that is a kidney function near the normal one, to 5, that is the last stage of kidney disease. This is very important too because it allows the physicians to know how to treat your kidney disease. Kidney disease usually progresses silently and you can lose most of your kidney function before you can develop symptoms. The last stage of kidney disease, or it is called kidney failure, and people with kidney failure need treatment in the form of dialysis or kidney transplantation. Unfortunately, most people don't qualify for a kidney transplant, or they have to wait a long time to get one. That's why people is put on dialysis. That is a treatment that is done several times a week, and it's a treatment that aims to replace most of your kidney function. However, dialysis is associated with a poor quality of life and a significant economic burden for the healthcare system. There are over 2 million people worldwide that are receiving treatments right now in the form of dialysis or kidney transplant. This is especially important in Manitoba because Manitobans have the highest rates of kidney failure in Canada. In 2019, there were more than 80,000 Manitobans receiving dialysis. However, if CKD or chronic kidney disease is caught early through screening, the risk of progression from chronic kidney disease to kidney failure can be reduced through changes in lifestyle and through medications. That's how in 2020, a person living with kidney disease can utilize resources that wouldn't have been available without scientific research. Recently, advances in kidney disease include accessible tests that allow us to diagnose chronic kidney disease in its early stages, a kidney risk equation that predicts the risk of progression to kidney failure, and medications that have dramatically slowed the progression of chronic kidney disease. Before 1960, a patient with kidney failure was very often facing a disease that was fatal. With the introduction of dialysis machine, it brought hope to patients that need renal replacement therapy. Not only that, but the research in dialysis treatment itself had advanced a lot. Before, the treatment was only done in the hospital. Patients needed to come 3 times a week for 4 hours each time. Nowadays, a patient can do the treatment at home. The first successful organ transplantation was a kidney in Boston in 1954. Kidneys are the most commonly transplanted organ nowadays. We also have a deeper understanding of the physiological, environmental, and behavioral risk factors for kidney disease. However, there is still much to be known about kidney disease, and that's the main goal of our research. In our lab where I work, we do research and develop and implement strategies for advancing screening and identify those patients before they reach the healthcare system. We also work in ensuring that individuals receive the most appropriate care in the most appropriate place to prevent the progression of the disease. And in patients who have chronic kidney disease, we do research on treatments that might improve the quality of care and the quality of life of these patients. As I mentioned before, chronic kidney disease population continues to grow, and these patients have a higher risk of cardiovascular disease and mortality. Many therapies for this group of patients remain unproven, and there is a need for clinical trials. Successful research for this patient population requires a collaboration between investigators, healthcare providers, patients, industry, and government. Research opens the door to a better future for patients with chronic kidney disease and for their families. Research in kidney disease have come a very long way and still have a very long way to go. That's why it is so exciting to work in this area. Thank you.
Hi, my name is Vivianne. I'm a PhD candidate at the College of Dentistry of the University of Manitoba, and I study the role of taste receptors and the oral microbes in the development of tooth decay in young children. So in the next few minutes, I will be talking about the association between taste receptors, the oral microbes, and tooth decay. And let's start with taste. So there are 5 different types of taste that humans can identify, and among those, sweet, umami, and bitter are sensed through taste receptors. And my research focused on the bitter taste receptors, or type 2 receptors, or T2Rs for short. And we know that sensing bitterness is very important for humans because most of the toxins they taste very bitter. And it's also important for us to identify the differences between bitter compounds and nutritionally important substances like sugar. Therefore, it's not surprising that there are 25 different bitter taste receptors and only 3 sweet and umami taste receptors. And but what are T2Rs? They are proteins and they are found not only on the tongue but throughout the body. And they are now considered to be chemical sensing receptor because they have many other roles apart from taste sensation. For example, in the lungs, taste receptors they can detect bitter compounds, including those from cigarette smoke, and they cause a protective reflex. In the gut, for example, they are responsible for the stimulation of the release of certain peptides that can tell the stomach wall how hard it should be contracting. It can tell the intestinal cells how well they should be absorbing the nutrients, and they can tell the brain how full we feel. So it's very important to investigate and to understand the different roles of bitter taste receptors throughout the different tissues. But now let's talk a little bit about the oral microbes. We know that there are more than 700 different species of bacteria and more than 100 different species of fungi in our mouth. And usually these oral microbial communities, they are highly dynamic and they often exist in equilibrium with the host and with the environment. But sometimes this scenario changes, and some bacterial and fungal species will cause the development of oral diseases, such as tooth decay. And tooth decay refers to the damage of the tooth structure that is caused by the acids produced by bacteria when they break down sugar in our mouth. And high consumption of sugar is linked to high activity of tooth decay. So there is a clear association between the oral microbes and their metabolism in tooth decay development. But what does taste receptor has to do with this? Why am I focusing on taste receptors? Well, one interesting thing about bacteria is that they can talk to each other and they can talk to their host. So the bacteria produce some molecules that are used for their communication, and human taste receptors can detect those molecules. So for example, there are some studies done in the upper respiratory epithelium that shows that one specific member of the T2R family, the T2R38, they can be activated by bacterial molecules, and they are responsible for initiating a defensive response. So for example, when those bacterial molecules activate T2R38, they cause an increased release of antimicrobial peptides that causes the killing of bacteria. However, for that to happen, the T2R38 has to be functional. And we know that T2R38 functionality is changed by some variations in the gene that encodes the T2R38 proteins. So variations in the genes could lead to variations in the protein, and that could lead to the functional or the non-functional form of the T2R38 receptor. But why is this important? Because if you have the non-functional form, your taste preferences could be affected because you wouldn't be able to detect some bitter tasting substances, and you would not be able to detect those microbial products there, and that response, the initial response against those microbial infections would not happen. So the scientists developed a simple test to identify whether the person has the functional or the non-functional form of this T2R38 receptor, and this is called the PTC paper test. And it is a simple strip of paper that has very small concentration of the PTC substance, which tastes very bitter. And only people with the functional form of the receptor will be able to taste this PTC substance. So when you put this on your tongue for a few seconds, you'll be able to tell whether you are tasting the PTC substance or not. People who are very sensitive to bitter tasting food, they are called the super tasters. And people with the non-functional form of the T2R38, they are not able to taste some of those bitter tasting substances, and they are called the non-tasters. But why is it important to know whether you are a non-taster or a super taster? Well, first of all, this can affect your ability to taste some bitter tasting food. This could lead to, if you're a super taster, you are very sensitive to bitter tasting and to strong taste in general, so you would be more likely to avoid eating the healthy bitter tasting vegetables. On the other hand, if you are a non-taster, you tend to put more spice and sugar on your food and drinks, so you'll be consuming more sugar and you'd be more prone to diabetes and obesity and tooth decay. So it's also important to know whether you're a super taster or a non-taster, because this can have an effect on your ability to detect those microbial products that I was mentioning before, and therefore it could affect your ability to fight infection. So for example, there are a few studies showing that people with the non-taster profile, the non-functional T2R38, they are more likely to have the more severe forms of upper respiratory infections. And based on that, we decided to look at whether the same thing would be true for tooth decay, whether having variations, and not only on T2R38 but all 25 bitter taste receptors and the sweet and umami taste receptor as well, having this variation, we want to know whether having these variations in these taste receptors could affect the composition of the oral microbiome, whether this could affect the taste preferences of the young children that we have in our study, and whether all of these could be associated with their risk of developing tooth decay. And hopefully with this knowledge, in the future we'll be able to develop a more personalized treatment and preventative strategies to prevent tooth decay in young children. Thank you very much for your attention.